The overall goal of this procedure is to produce high yields of fully glycosylated and enzymatically active granzymes in human embryonic kidney 293 T cells. This is accomplished by first expanding HEC293 T cells in appropriate tissue culture dishes. The usual preparation size after the expansion consists of 20 to 25 culture dishes. The second step is to transiently transfect the expanded cells with a Granzyme expression plasmid using the cost-efficient calcium phosphate method. Next, the cell supernatants are harvested, and the secreted Granzymes are purified by immobilized metal affinity chromatography. The final steps are to activate the Granzymes by enterokinase treatment and to further purify the proteases via cation exchange chromatography. Ultimately, enzymatic activity is tested using a rapid and reliable colorimetric assay.
As the availability of active protease in sufficient amounts is a limiting factor in Granzyme research, this method can help answer key questions in the field, such as characterization of new cell death pathways in different organisms, or identification of novel protease substrates in whole proteome approaches.
To express Granzymes, first grow HEC293 T cells in 20 milliliters of culture medium using 150 by 25 millimeter tissue culture dishes. Split the cells at 80% confluency. Cells loosely